Welcome, everybody. Thank you for coming tonight. I'm Jay Millius from the biology department, and it is my pleasure tonight to introduce Dr. Mark Ptashne. As quoted by Joe Sandbrook in the book Inspiring Science, Jim Watson and the Age of DNA, Ptashne's successful search for and characterization of the elusive repressor of bacteriophage lambda, work that spanned 2 decades, can fairly be regarded as the greatest sustained experiment of the last century. Indeed, Mother Nature does not give up her secrets easily, and Dr. Ptashne's dogged efforts have profound implications for the field of genetics and for biology. The significance of Dr. Ptashne's work is that he described a genetic switch, how genes are turned on and turned off on a molecular level. The bacteriophage lambda, a virus that infects bacteria, can replicate its DNA by 2 different mechanisms. The bacteriophage DNA can either insert into the bacterial genome, and then it will multiply when the genome is copied as the cell divides, or the bacteriophage can copy itself inside the cell, producing approximately 100 progeny, then bursting out of the bacterium, killing it. Dr. Ptashne purified the lambda repressor and showed that it binds to lambda DNA. Repressor binding provides the explanation for how the decision, this switch, is made. Dr. Ptashne went on to thoroughly characterize the system, identifying the regulatory protein CRO that also recognized the lambda operators, describing positive regulation and feedback loops. He essentially founded the field of gene regulation. Genetic switches are common in the biological world. They are, for example, the key to how cells respond to environmental signals, metabolize specific nutrients, develop into different tissues, form macromolecular structures, lose control of cell division, leading to cancer, to name a few. Quoting Dr. Ptashne from a recent interview published in PLOS Genetics, the more we learn about gene regulation and development in higher organisms, the more it seems that lambda teaches us. Thus, he provided us with a model, a framework for how these and many other genetic decisions might occur on the molecular level. Dr. Ptashne received his BA in chemistry from Reed College in 1961. He received his PhD from Harvard University and was a faculty member at that institution from 1968 to 1997. Currently, he is a Ludwig Professor of Molecular Biology at the Sloan Kettering Institute in New York. Among many research articles, Dr. Ptashne has written 2 books on this subject, A Genetic Switch, from Cell Press and Blackwell Scientific Publications in 1992, and with Alex Gann, Genes and Signals, Cold Springs Harbor Laboratory Press 2002. And I seem to remember some books out there on the table on the way in here. So he received the prestigious Lasker Award for basic research in 1997, and that is considered really only second to the Nobel Prize. But Dr. Ptashne is also a talented violinist who studies at the New England Conservatory of Music. He was a founding member of Reed's Collegium Musicum. Dr. Ptashne is the recipient of this year's Thomas Lamb Eliot Award. Here to present the award to Mark is John Kroger, president of Reed College.
So the Eliot Award lecture is one of my favorite events every year here at Reed. The Eliot Award is given to a graduate of the college who most exemplifies the virtues that we care about in the Reed community. And there's really no one who exemplifies intellectual rigor and creativity quite like Mark Ptashne. I have to say, it's actually not true that the Lasker is considered second to the Nobel Prize. It's actually considered second to the Reed College Eliot Award in terms of its distinctions. So, Mark, you'll be able to put that behind you after tonight, which is very exciting. Mark, I was sitting next to him when Professor Melly's quoting, a scientific history, said that his work represents the most important sustained research project of the last century, lasting 20 years. And he immediately turned to me and said, "That makes me sound like a very boring fellow." The 2 things I would tell you: one, I've really enjoyed having some time to spend with Mark over the last few years. He's very passionate about the value of Reed and Reed science education. He's been very helpful to me in thinking about ways we can continue to sustain our commitment to excellence. He's also a great violinist, and I've just confirmed with him that he's willing to come back to Reed if invited to play the violin next time. And Mark, if you do that, we'll probably try to corral you into spending some time with our biology students as well. So, ladies and gentlemen, the last few recipients of the Eliot Award have been remarkable people. Arlene Bloom, the pioneering mountaineer, Gary Snyder, the great American poet, Richard Danzig, the U.S. Secretary of the Navy. And it is really a pleasure tonight to give the award to one of the world's greatest scientists and researchers. So with that, Mark, please come on up. And let's all give Mark Ptashne a hand.
Whoa. Wow, this is quite exciting. Actually, the most amazing thing about it is that how many people would have thought that when I returned to Reed, I'd be wearing a tie? So I'll see if I can proceed without ripping it off. Just sitting there, I thought, gosh, I should say a word or two about, wait a minute, how do you get this going? You click down here, but I don't see the thing. Okay, that'll do. I got it. So I just thought I should say that when I was at Reed, there was a fly Drosophila geneticist, okay, named Tahir Rizki, a real Pakistani with a beard and the whole thing. One day, Tahir brought me into his lab, and he said, "Look at this." And I looked at it. He says, "That's the result of a reciprocal cross." A reciprocal cross. Remember that? And so somehow, he sent me to the great Ed Novitsky, who was a famous chromosomal mechanics man at University of Oregon, a Drosophila geneticist of great renown and wit. We actually spent a couple of summers at Crested Butte, Colorado, where all the great fly people came, including, by the way, I just remembered, H.J. Muller. So then Ed, having a famously wicked sense of humor, one of his things was he had a good friend come give a talk, and he designed an electronic gizmo. He could do this, so that the clock ran slowly. So he hated Novik and Stahl because they were the founders of molecular biology at Oregon. And he figured he'd send me, the Trojan horse, to destroy them. So I went to their lab and almost destroyed him. But then Stahl had just come back from the famous Melselssohn-Stahl experiment, and he said, "We got to get back at this guy Melselssohn, so why don't you go work with Melselssohn?" He was at Caltech. I went to visit Caltech, but Jim Watson had just visited Matt and said, "You got to come to Harvard," so I went to Harvard. So that's the end of my everything, all boring. Right, so actually, you know, I remember reading, now that you mentioned it, this guy who won the Nobel Prize in chemistry, and he said the reason he did so well was that he had this, what do you call it, attention deficit disorder? So he switched subjects every couple days until something worked. And so I can see I'm on the wrong path here. All right, so this is a curious situation because I'm never going to use the word repressor. I think it was all misleading. We'll see. I may have to explain that later. I'm going to try not to get into it at the moment, but see if what I say makes any sense. So about the time I was finishing up at Reed, it was already clear that there was this pair of problems called the Evo-Devo problem, evolution and development. During development, you know, your genes don't change. You don't get new genes. You don't get rid of old genes. And during evolution, it turns out, certainly all the kind of evolution Darwin knew about, there may be occasionally some new genes, but mostly it's the same old genes. What we have is what a chipmunk has. So why are we different? Well, obviously, it's something to do with regulation. And that'll maybe become clear as I go along. So how do we think about that problem? Well, modern day science would look at this kind of thing and say, no problem, we'll just sequence everything. But about this time, there was a huge commotion coming from the Institut Pasteur my then and continued heroes, actually. I feel like even though I never met Monod, I met Jacob once, never met Leboeuf, who's, let's see, that's Jacob there, that's Leboeuf, that's Monod. It's as though I had conversations with him every day for about 40, 50 years, still do. And when students and professors say, I never heard of Jacob Monod, I say it's like trying to do modern physics and never having heard of Newton or Maxwell, whatever Maxwell did, I don't know. But anyway, it's not the historical fact of paying them homage. It's when great intellects come along and make a field, you've got to come to grips with how they thought it through. And these were... Anyway, so that's why I think Reed has an important role to play. Now, what was so exciting about what they did? In a word, let's greatly simplify, but let's just say they did this. They got a mutation, meaning they knocked out the functioning of a specific gene, and that gene regulated the expression of a different gene. So something that was on only under certain circumstances, when you gave it sugar, for example, would be on all the time in the presence of this mutant. A different mutant would affect some other regulatory process, for example, the lambda thing that was just mentioned. So the key issue in retrospect, and you always have to look in retrospect, you never know. This is a really important point to make. In retrospect, the important thing was specificity. How do you tell one gene from another? They all look alike. There's boring pieces of DNA. So how could something tell a gene to be expressed or not? In particular, now that you had a mutant, you knew something existed that did this. It's a genetic experiment, so you could always, but anyway. And so the question, okay. So I got a lot of slightly pretentious quotes here. I don't read Goethe, but this comes from an absolutely magnificent page I saw. I had to cut it way down, but this is true. I mean, you thought, oh my God, there's a mutation that affects a specific gene. Maybe we can explain the whole universe if we could understand that little thing. And blah, 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 blah. So the issue was, what did those genes, that regulatory gene and other genes like it, encode, and what did they do? Crick said, in solving scientific problems, it is almost impossible to avoid falling into error. Typical English. And Crick, of course, was a magnificent intellect. And so he said, intellectual collaboration helps jolt one out of false assumptions. Something to keep in mind. So I was lucky in various ways. One way was that in following Watson and Messelson and so on to Harvard, I came across this guy, Wally Gilbert, who maybe you've heard of, who was a physicist and who was just going into molecular biology. And so we set out on this kind of, in some circles, famous competition. I actually learned more from Wally than I learned from anybody. He was trying to destroy me, but he was so generous that anyway, so I'm not, if you want to talk about that, we can, but this is a rare thing when you have a really serious person who will look you in the face every day and say, what you're saying is total bullshit. Nowadays, my God, if you said something like that, you go directly to jail. This is a big problem, don't you think, in education? Okay, so it turned out, huffing and puffing, we isolated the products of two of these regulatory genes, and guess what? They were proteins. Now that was an amazing thing, because how could a protein tell one gene from another? If it were, for example, an RNA molecule, it could just open the DNA and pair with the thing. But they weren't, they were just pure proteins. And the next discovery was that they not only genetically do they do this, but biochemically, you can show that they select out the sequence that they should bind to, and they bind there. Now, I put bind in quotation marks because this is a tricky thing. In this, we're talking here about interactions in the entire most of everything I'm going to talk about. They're like this. They're not like locks and keys. This is a very hard thing to come to terms with. The physicists call this a dynamic interaction. So you'll see this haunts all discussions, and until one faces up to it in some way or another. Okay, so but let's go back to the very basics. How could a protein possibly tell one sequence from another? God arranged it in the following way. An alpha helix, which, proteins look like spaghetti, but they often have alpha helices in there. An alpha helix, which can sit on the outside of a protein, can fit into a major groove of DNA and make certain contacts with edges of base pairs. That must be the way it works. Problem solved. There's only one problem. That alpha helix can see maybe 3 or 4 base pairs. Now, if you consider the vast number of base pairs in even a lambda genome, a viral genome, or an E. coli genome, human genome, can't possibly be specific enough, and it's not tight enough so that a single one of these helices might take a quick look and it'd be gone soon, or it'd be elsewhere. So I'm going to now I don't know if you react to this the way I have for the last many years. Another intellect that really amazed me is this guy, H.A. Simon, who won the Nobel Prize years ago for very abstruse stuff. I have no idea, computer stuff. And he wrote this magnificent little book called Sciences of the Artificial. It went through 3 editions, just like Genetic Switch, by the way. 92 was the 3rd edition. And he said these kind of weird things. Engineering, medicine, architecture, painting, a lot of other things are concerned not with necessity, but with the contingent. In short, with design, by which I think he meant, I guess he must have meant, that you're starting somewhere kind of arbitrary, and now you're stuck with that. Now you have to make it work. And so he said one of the problems in design, if you go to one of these schools that teaches architecture and molecular biology and all that under one roof, would be how to build a system, a reliable system, from unreliable parts. But of course, that's just our problem, right? That alpha helix in the major group is a very unreliable way to do anything. So then you think, oh my God, this great intellect is going to fill us with equations, and how the modern systems biologists are. By the way, I've asked a few, they never heard of Simon, so it's amazing. Anyway, maybe because he said this, which is devastating if you're a professional. The requirements of design can be met by an adaptation of ordinary declarative logic. Really? That means just you and I can think about this problem. We don't have to get a license from the... Oof. Okay, so here's the answer, and it makes perfect sense, and all you need are balls and sticks to understand this. There's a phenomenon called cooperativity. Everywhere in the biological universe, and the thing I want to emphasize right now is that cooperativity is what increases specificity, and it enables you to use these unreliable parts, putting them together to make something reliable. So what do I mean? Well, here's how cooperativity works. Imagine you had 2 of these alpha helices, one here and then another one here, and they're connected by a loop. Now, the unreliability means that one of them will tend to come off a lot, right? But at that billionth of an instant, this guy will tend to be there, and of course, vice versa. So simply by linking these guys, it's intuitively completely obvious, I guess, too simple for sophisticated scientists maybe, you increase the stability and the reliability of the binding. It's a way you can use a very unreliable element to get more reliable. And this is, it turns out, what nature does over and over again. For example, let's say in this particular case where it's of no significance really that there are 2 domains, it's just a holdover. This particular protein has a carboxyl domain. I mean, it doesn't matter. This is the part that recognizes DNA. So now you need to invent a protein-protein interaction between these guys to make a dimer. And then the dimer can bind to DNA, and now 2 alpha helices are touching, right? And we have just that picture. If one lets go, the other doesn't. But you see you're not satisfied? Okay, so then you add another site, and now you get, whoops. Wait a minute. Yeah. Then you get this sort of thing. So now you've got a protein-protein interaction here, a protein-protein interaction here, and DNA-protein interaction here. And this sequence is now much more unique than one of these little teeny ones. You got 4, basically. And the probability that you'll find 4 close together is very small. So, and this you see over and over again. You take these simple reactions and you reiterate them, and you link them, and you make them more and more reliable. So the way you make a specific protein-DNA interaction is by both designing the protein to recognize the DNA, but also by evolving these protein-protein interactions, most of which, by the way, are quite weak. This is another thing, this is something that makes, I used to say, and this got me in a lot of trouble, this is why chemists can't do molecular biology. The interaction between these guys is like a factor of 10, a kilocalorie. That doesn't register on a proper person's scale, right? So how would you possibly measure it and know it's true and so on? Well, that's another whole story, but you do it by a combination of genetics and biochemistry and logic, but that's how it works. It's very difficult to just look and see this. It requires a whole... Okay, there's one other point that I think I'm just not going to mention, except to mention it, and that is that these specificity reactions depend on concentrations. So can we come back to that? Because it has, I mean, if you want to talk about it, because, okay. Another philosopher of science, Nietzsche, said, because after all, you could say, well, so what? You've discovered how a protein can bind to DNA in bacteria, who cares? He says, a stroke of luck. Science discovers things that furnish the basis for further discoveries. After all, it could be otherwise, which I particularly love. And of course, it could be. So ye of little faith will never get by doing science, because you see what I mean. And so it turns out that, yes, Nietzsche, you're right. Cooperativity not only explains how you get specificity out of protein-DNA interactions, it also explains how protein can activate gene expression. The word activate is totally misleading. All of our language about this actually is misleading. But the fact is that you can show in various ways, genetically and biochemically, if you add this protein and it binds to DNA near a gene, transcription, copying into RNA, goes up. So that thing's called an activator. The word implies that you stick a key in a lock and, activate something like a car. But that's not right at all. It's just another example of cooperativity. So an activator, you see, turns out, binds DNA. There should be several of these sites here, it doesn't matter. With one surface touches DNA, and with another surface touches this enzyme that transcribes the gene. And just as in the illustration I gave you before, one protein stabilizes the binding of the other, so this activator stabilizes the binding of this enzyme. And so now this just happens a lot more often than it otherwise would happen. Science, in a way, can be quite disillusioning, because you've got all these fancy ideas, activation and blah, blah, and be a scientist, and it turns out it's just this little bit of Velcro, that little factor of 10, it's like nothing. It's not my fault. So let's look at the type activator. It's got 2 surfaces, one for DNA binding, and one for, it's called the activating region, which touches RNA polymerase when it's appropriately bound. And the important result early on was, before we really knew a lot of things, is that if you mutate this sequence, the protein binds perfectly normally but does not activate. So DNA binding, and of course there's a whole slew of models that say, well, the way you bind DNA and it huffs and puffs and so on, has nothing to do with anything. It's just bringing the activating region where it can touch polymerase. On the other hand, if you mutate the DNA binding domain region, the activating region can't do anything. It's just floating around. Okay, so it's important to abstract that so that you understand that this is a gizmo that has two surfaces that are independent in the sense that they can be independently mutated. And one has an effect only in the present, only when connected to the other. And I don't know, we could go on a lot about this, but it's the world's dumbest, simplest reaction. Activating regions in bacteria have all kinds of different sequences because they tend to bind here, 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 here. And then when they touch polymerase, they touch different surfaces. It doesn't matter because all it is a cooperativity reaction. It's what we call recruitment. We say that the activator recruits the polymerase, and any way it can touch polymerase, it will recruit it. This is, for example, just so we don't get confused later, here's the famous lambda repressor case. Now, this is a real problem. It was called lambda repressor, and in fact, Jacob and Monod believed the world was run by repressors, things that turn genes off. And for the moment, I'm just ignoring that because it's not the fundamental thing. They got that backwards, but this, don't worry about it. They taught us how to think, and they got almost everything else right. So anyway, so here's this protein working as an activator in its guise as an activator. DNA binding and activator, they happen to be on the same domain. I'm just making that point. This domain, the carboxyl domain, just as in that picture, just helps them dimerize and so on. So already, life is beginning to resemble, not an erector set, but what do you call those things? Tinker toys or whatever. Right. Any activator can activate any gene. You just have to have its binding site. Its binding site, every activator has a different binding site. You just have to have those nucleotides near the gene so that the activator can bind there. So already evolution is, this is ridiculous. And you're going to see it gets easier when we get to eukaryotes. Why am I showing you this again? I don't know. All right, so remember we said we are trying to understand this problem, the Evo-Devo problem. So let's start with development. So what is development? We have to establish a state of gene expression, and then we have to remember it. We have to maintain it. And so there are two phases to all examples of development: establishment and maintenance. Here's the establishment phase that you've already seen, which, good luck. So let's see what Simon has to say. The central task of a natural science is to make the wonderful commonplace, to show that complexity, correctly viewed as only a mask for simplicity, to find pattern hidden in apparent chaos. Well, it's easy for him to say. Now what? Well, here's his suggestion. One way to solve a complex problem is to reduce it to a problem previously solved, to show what steps led from the earlier solution to a solution of the new problem. So let's take a case of development. It's one of the ones that Jacob Monod originally, in a way, talked about, or did definitely talk about. And it has to do with this virus lambda, which injects its DNA into a cell. And here there are there's a, I'm sorry for these names, forget them. There's a piece of DNA, this is about 2 genes out of 40 genes. And here's what happens. The first thing that happens is this gene is transcribed. Now it's unusual in that it doesn't need an activator, simply because the polymerase binds quite tightly to it on its own. That's very unusual. I think it's non-existent in eukaryotes and it's rare in bacteria, but in viruses you might find it. Okay, so what does that thing make? Well, it makes a protein called C2, which is what? It's an activator. And it binds, its site lies here. And it therefore turns on a transcript that goes to the left. And in so doing, it produces this thing, which we just saw the picture of, the famous lambda repressor. And now lambda repressor binds here. And so we're gotten through the establishment phase because, let me see, what's the next slide? Because Maintenance, the way you maintain this state, is by positive feedback. Meaning, the repressor, this protein, the activator, its site is put here, 2 of them, so it feeds back and keeps itself on. Simplest thing in the world. And it doesn't, so we've got a tail of 2 activators, C2 and C1, and just by putting their sites in the correct places, that's all we have to know. And the system works, and it's self-perpetuating. If you want to see a fancier version, it looks like that, and all the protein-protein interactions are shown here. There's very weak ones here, here, and of course here to recruit the thing. Now this thing is made, feeds back, and just keeps it on. So as the cells divide, the key molecule, C1, is distributed to the daughters, and they just maintain their state. And this thing is incredibly stable, turns out. It just goes on forever until there's a signal. And a signal destroys the C1 and causes another phase of gene expression, but we don't have to worry about that. What I just want to emphasize is that it's like child's play, given what we know about how activators work, to arrange a system with a couple of activators and a positive feedback loop, very easy to maintain, to construct. It's a property of the system because it binds. And we've done maintenance, and we've done establishment and maintenance, and we've made an incredibly stable thing. Is that clear? Please interrupt me if, let's see what Simon has to say. The task of natural science is to make the wonderful commonplace, to show that complexity is only a mask for simplicity, to find pattern hidden in apparent chaos. Sounds like we're getting there. So now we have this problem. This and tons of other stuff is about bacteria. But of course, the professionals will tell you, poof, you haven't sequenced the human genome. How do you know it has anything to do with humans? Fortunately, there's an organism called yeast. And yeast inside of a yeast looks just like the inside of a human cell, in the sense that the DNA is all wrapped up in these things called nucleosomes. There's a separate nucleus, all kinds of things. I mean, yeast is a genuine, as they say, eukaryote. And the great thing is, because of all the experiments that these wonderful people have done, there's a little vodka in here, by the way, so woohoo. And since you can grow yeast almost as easily as bacteria, and you can get mutants, you put them on plates, you can do all this stuff, you could actually go in and ask, well, how does any of this apply to yeast? And fortunately, actually, the single statement I love above all else, basic models tell us more than we can at first know. You don't just know it when you look at the model. Years go by, and then I see. That's what that was telling us. But, it's a constant. It's like the ring, right? What happens in the ring? Some of you know the ring, right? They do it in Portland, don't they? Right. So every couple days, somebody stops and tells the whole story all over again. And I think about it, what it means. Well, that's sort of what science is like, or should be like, but often isn't. So others had by now shown that there was a set of genes in yeast that was totally off if you mutated a gene called GAL4. Now, it's no secret, GAL4 is a protein, by now you can tell that easily enough. And the surprising thing that I'll just leap ahead to, which makes all of the arguments easier, is that if you put the sites to which GAL4 binds, you put that DNA site in any other organism that we know of, any other eukaryote, plants, flies, worms, anything, and you express GAL4, GAL4 will land there and turn on the nearby gene. So whatever GAL4 does, it does universally. It sounds like what I just said about bacteria, right? The bacterial activators will activate any gene if they can bind near it, And so that's turning out to be true of Gal4, which is kind of astonishing. So now let's just remember, what was the formal property of the activator that we were so entranced by? Namely, that it has two important surfaces, a DNA binding surface and an activating region. And it turns out that Gal4, although I'm showing it, Gal4 also has two surfaces, and it's much easier to study in eukaryotes because everything is cruder. So it was arranged that the activating region would be on a different domain, so you can just cut you can cut the protein in two, and you know right away. The bottom part binds to DNA as a little glumph. The top part is an activating region, doesn't do anything. Unless, and I had a whole bunch of things, but maybe just grasp this idea. If you attach the top part of Gal4 to any DNA binding domain, it will work as an activator. If the DNA binding site is near a gene. So you make it in yeast, you make it in plants, you make it in flies, boom, it activates. Now, how does it activate? Well, let me just say that in fact, eukaryotes are so crude that you can make perfectly good activators by just junk that's lying around the kitchen. All you need is some DNA binding domain, and now you can just add random sequences. And a lot of them work. Not all of them, but a lot of them. Very amazing, actually. Nature just did not have to worry about this problem. The only problem was concentrations, come back to that, and making the gizmos go to the right place. So how does it actually work? Well, it works, I've got to say, this is really still an astounding thing that this general picture must be right, but take it or leave it. So to transcribe a gene in eukaryotes, you need like 100 proteins. It's depending on your worldview, a nightmare or a dream, because you can get endless. And so what happens is the activators bind, and somehow, touching various parts of this huge thing, recruit them, just like the bacterial activator did. Except that in this case, it's a bunch of guys binding cooperatively. Now, how do we know it's recruitment? Well, there are a lot of ways, in fact. But one way is, just as in bacteria, you don't need an activating region. You need just something that contacts part of the machinery, some arbitrary protein-protein interaction. Boom, it works. That's true in bacteria, too, something we actually first realized in yeast, and then Anne Hochschild did that in bacteria. And then for those of you who know what the two-hybrid system is and all these things, they work because the whole system is so crude. Right? The only German I know with a really good sense of humor is Oliver Hobart. Anyway, he works on sensory neurons and C. elegans, amongst other things. That's a worm, a disgusting little. And so the question is, how do you establish and maintain a sensory neuron in a worm? Well, the first phase is, you've seen this before, it doesn't matter. But the good news is, you're willing to hear now that it's complicated, but it's not mysterious, right? You know who said that? The immortal Homer Kelly in his famous book, The Golfing Machine. And he was familiar with the book? Believe me, you'll never hit a ball again if you read this book, but it's the great classic in various ways a club can come at the ball. So what happens? Well, during establishment, there's a huge huff and a puff, and developmental biologists will be worrying about that for a long time. And you make a certain activator. The activator binds to the blue site, which I'm calling an enhancer element. Okay, now that turns on the key 1 gene, which is itself an activator. And guess what? The activator feeds back, binds to the maintenance enhancer, keeps itself on, and then it goes around turning on all the genes you need for being a sensory neuron. It's exactly the way lambda works, except that you're using, let me not say that. If you're interested, we can talk about enhancers versus the other thing, but the principle is the same. Wow, I'm almost done. Great. Because now I want to say something really, It's obviously true that what we learn from transcription applies throughout the molecular biological world wherever you deal with an enzyme that works on another macromolecule. Right? So the way enzymes work that work on little teeny molecules is they have an active site, and the thing goes in the active site, and if it's slightly different, it can't go in, and everything's fine. But that's obviously not how kinases and phosphatases and splicing enzymes and ubiquitilators, everything else doesn't work that way. There's one enzyme, and so, okay, let me set you up for this by saying this. We talk about gene regulation. That's wildly misleading because the genes aren't regulated. What's regulated is RNA polymerase. What the activators do is impose a choice on the enzyme as to which gene to transcribe. We call it imposing substrate choice by recruiting it here or recruiting it here. Obviously, that's what's going on. And it turns out that this is a very general rule for all these enzymes. For example, it's unbelievable that it took me to figure this out. Ubiquitination, everyone's known for years that there's this enzyme called E2, right? Big monster that puts ubiquitin on proteins. And they then go, whatever happens to them. And then everyone knew, thanks to what's his name at Caltech, that you had to add something, that there was something called an activator, and a ubiquitin ligase, which you threw in the mixture, and now the reaction would go, and a bunch of proteins would get ubiquitinated. But because it was called a ligase and an activator and so on, it's completely confused. But what actually is happening is that the E3, which is an activator, is simply a recruiter. With one surface, it binds the target, and with one surface, it binds the enzyme. And another E3 will take it to a different enzyme and to a different target and a different enzyme. The generality is that, the generality is that all of these enzymes have specificity. They are specific in their active sites. A kinase, after all, can only add a phosphate. And there are two kinds, three, I don't know, two or three. Anyway, tyrosine, blah, blah. But then they've got all these proteins to choose from. How do they know which one to do it on? And the answer is that there are residues outside the active site which impose specificity, either by directly binding or by intermediates recruiting them or by scaffolds. It's a very general thing. And the scary part about that is, the scary thing is that it means the entire world of specificity is based on mass action. Gotcha on that one, huh? So imagine curves and equations and so on. So you have these signal transduction pathways. So how does the kinase know to go to this guy and not to this guy? Well, okay. The reason the world is so complicated is that that's a hard thing to do. You have to have things that block it going to the wrong place. You have to have recruiters that blah, blah, blah. And so these binding reactions, just like a protein binding the right side on DNA, have to determine the specificity. Is that clear? This is, I hope, if you think about this, you'll find it very scary. Because obviously it's what goes wrong in a lot of cases, like cancer. Remember I mentioned concentrations? Well, the difference between binding to a correct site, maybe with the help of a cooperatively recruiting guy, and binding to an incorrect site is at the most, let's say, a kilocalorie, factor of 10. So similarly, what if you overproduce a kinase, a factor of 10? That happens all the time in cancer. It goes to the wrong site. And if you look, the wrong substrate. And if you look at any of these papers, these guys always say, it's quite amazing, blah, blah, and now we got a resistant mutant, and now it's regressed, and they got the tumor again. And they only mention if you really pry, oh yeah, by the way, that kinase is overproduced 100-fold or 10-fold. The amazing thing to me is not that it goes wrong and that we age, well, whatever that is. The amazing thing is that it ever works in the first place. And what you can see, I think, the reason molecular biology can be so tedious is that when anyone examines any one of these things in great detail, there are all these little gizmos that help. Do you want me to give you one more about this? Okay, because I'm really obsessed about this. All of these enzymes, most of them, there are a couple, you can get around this, I know, but basically, like polymerase and so on, they're all constitutively active. So that means they can work in two ways. They can either work when they're recruited, right? They work strongly, but they also work as part of the background. So there are two things, of course, then. If they get overproduced, the background becomes the foreground. But the second thing is, I don't think this is appreciated enough. The second thing is that everywhere there are basal levels of these things, and everywhere nature, evolution has had to stand on its head to make sure the basal levels don't screw things up. Okay, so for example, transcription. Remember I mentioned that there are, I guess I can say this now. Remember I mentioned that there are nucleosomes. All the DNA is wrapped up in these forbidding structures. Well, the main thing that does is to keep the basal expression, the unrecruited action of polymerase, very low. We know that if you screw up the nucleosomes, you'll start to get this background gobbledygook. There are various kinds of, kinases, for example. They're always working at a background level, and if you inhibit the phosphatase, the major phosphatase, all the signal transduction pathways turn on without signals. Ubiquitinating systems. There are, of course, deubiquitinating enzymes, and then there are added tricks, like if the thing bumps in and makes a little ubiquitin chain, it doesn't do much. But if you recruit it and you want it to work, boom, it makes a long chain, and now you get whatever ubiquitin does. So I think everywhere you know what ubiquitin does, huh? Is that why you're laughing? No, not you, the lady behind you. Okay. I think I should have some more vodka here. So maybe that's the point. As I say, it's amazing it works in the first place. Okay, why don't I stop this rant and see if you have any questions? Thank you. You can applaud.